Hi everybody, we're going to pick up with nervous system. This time we're going to be talking about the central nervous system and the brain. Now we're going to cover the anatomy of the brain first, and then we'll dive into the physiological functions. That will be in a separate PowerPoint. So in your course pack, if you would like to follow me, I'm on page 111. We've talked in a previous PowerPoint about the protection of the central nervous system. The organs of the central nervous system include the brain and the spinal cord. We know that there is bone protection over both brain and spinal cord provided by the skull over the brain, and the vertebrae provide protection over the spinal cord. We also have talked about meninges or membranes that cover and protect those organs. So let's review those now. The meninges are basically connective tissue membranes, and they are the Dura Mater, Arachnoid Mater, and the Pia Mater. The dura is a tough outer layer, sometimes called the tough mother. Dura technically translates to tough or hard, matter to mother. The arachnoid matter is a middle layer that has a set of fluids that run through there called cerebrospinal fluids. We'll talk more about the circulation of CSF in just a few minutes. The deepest of the three layers is a very thin, delicate layer called the Pia Mater. It's the soft mother. And so this particular layer directly contacts either the brain or the spinal cord. This is a great image for us to study the layers around the brain and the spinal cord. You can see the skin, the periosteum of the bone, and that bone could be either the skull, which is actually what's shown here, or it could be the vertebrae. You can see the dura mater is anchored to the bone. It's also anchored to the underlying arachnoid matter. The arachnoid matter gets its name from a spider web-like appearance. It is through that spider web-like membrane that we see cerebrospinal fluid circulate around the outside surface of the brain and spinal cord. The deepest layer is Pia Mater, and like we've talked, that is a delicate layer that anchors itself directly to the underlying brain or spinal cord nervous tissue. Now, this picture shows us the back side of the skull. You can see the cerebellum down below. That's how I know that we're looking at the back side. On the right-hand side of the skull, you can see here there's not only bone that protects the brain, but you can also see the tough dura mater. It almost looks like wax paper in appearance. Something else you'll want to note about this picture are the large blood vessels traveling around the dura mater. So you can see them, they're called sinuses here. There's a transverse sinus and there's actually a superior sagittal sinus, both shown in this picture. Now, sinus is a name for a large vessel. This one happens to be a vein. These are not to be confused with the sinuses that may be found around your facial bones. Now, these sinuses lay in the dura mater, so they're wedged in the grooves between the brain. There is a longitudinal fissure between the left and right hemispheres. It is in that fissure that the superior sagittal sinus will lay. So this groove right through here between the left and right hemispheres is where we have the longitudinal fissure. On the horizontal axis here, right through here, we have another fissure called, well, the transverse fissure, and there is a transverse sinus housed within that one. To not be crude, but to give you a way to think of this, they're kind of like wedgies. These blood vessels in the dura mater are wedged into those fissures. So they lay on the outer surface of the dura mater, and they're actually collecting cerebrospinal fluid to be returned back to blood flow and circulation. Here again, you can see those three layers. You can see the outermost dura mater. You can see the fluid called cerebrospinal fluid. You can see the spider web-like arachnoid matter with its subarachnoid space. And you can see the deeper Pia Mater, which is directly connected to the brain or underlying tissue. In this view, you can see the two cerebral hemispheres of the brain, the left and the right hemisphere. The groove that runs between those two hemispheres is called the longitudinal fissure. It is in that fissure that we have a blood vessel that lays along that dura mater. Do you recall the name of that blood vessel? It's called the superior sagittal sinus. Here in the lateral view of the brain, you can also see the transverse fissure. It is found between the cerebrum and the cerebellum. So this groove that runs right through here is the one we're talking about. Do you recall the name of the blood vessel that lays here? It's called the transverse sinus. So let's talk about the formation of cerebrospinal fluid, sometimes abbreviated as CSF. Now, cerebrospinal fluid is formed from the blood vessels that flow into your brain. These are not the sinuses. These are actually blood vessels that flow into your brain, branching off of the aorta. Let's talk about what you see here. These are capillaries traveling through the brain. Lining the walls of those capillaries are specialized cells called ependymal cells. You'll notice those ependymal cells are ciliated. That's what all this outer structure is along here. Now, when we are working on getting cerebrospinal fluid into circulation, the vessels squeeze out and release fluids from the blood, but not the blood cells themselves. So we see only the fluids in the blood that contain filtrate, like glucose, oxygen, vitamins, and ions, being pushed out and circulated by those ependymal cells. At the same time, wastes and unnecessary solutes may be reabsorbed back into those capillaries. So what we don't see in this picture is that we're putting this cerebrospinal fluid into circulation around the outer surface of the brain and spinal cord, but we start off by being inside the brain in ventricles. These are some of the ventricles of the brain where choroid plexuses are established and set up the flow of cerebrospinal fluid. Let's talk about the ventricles that we see here. Now, there's a third ventricle, a fourth ventricle, and lateral ventricles. There are no first and second ventricles. So here we can see the lateral ventricles are actually housed in either hemisphere of the brain. They look almost like sheep horns. The third ventricle here is situated between the two hemispheres, and as we travel down, we go through an aqueduct called the cerebral aqueduct into a diamond-shaped fourth ventricle. Okay, so these ventricles are going to help us circulate the cerebrospinal fluid, and that's where this fluid is housed, is within these ventricles of the brain. As we continue down toward the spinal cord, that fluid can actually travel in the central canal. Now eventually this fluid will return to the outer surface and nourish and wash away things like waste products and solutes that are unneeded in the outer surface of both brain and spinal cord. So let's review. We have lateral ventricles in each hemisphere of the brain. We have a third ventricle between the two hemispheres. If you travel down a bit, you'll go through an aqueduct. You'll hit a diamond-shaped fourth ventricle. I know it doesn't look diamond here on the right-hand picture, but on the left one it does. And then we travel down into a central canal. So we're actually talking about circulation of cerebrospinal fluid, and we're going to establish CSF flow in choroid plexuses. So here's a few. There's a set here in this third ventricle area. There's a set of choroid plexuses in the fourth ventricle area as well. We're only going to pick on one of these and make it our starting point. And we're going to start on the one that sits here by the lateral ventricles. Actually, this one here, choroid plexus of the third ventricle, is where we're going to start our conversation. So when cerebrospinal fluid is made here at this area, it flows into the lateral ventricles in each hemisphere of our brain. It then will travel down through that interventricular foramen, which is a hole situated between these two ventricles. It travels down into the third ventricle, which we're looking at here, and then into the cerebral aqueduct. That cerebral aqueduct here is called mesencephalic aqueduct, but we'll call it the cerebral aqueduct. From there, we travel into the fourth ventricle. Recall that it has a somewhat diamond-shaped appearance in the other view. From there, we travel down into the central canal, and the central canal takes us into the spinal cord's interior. Now, I wanted to show you this picture because you'll notice at the tip end of the spinal cord around L1 or L2, that cerebrospinal fluid drips out, and now it travels in the outer surface of the brain and spinal cord. It now travels in the subarachnoid space that surrounds the outer surface of brain and spinal cord. When we reabsorb cerebrospinal fluid, we can pull it back into the sinuses laying in the Dura Mater. For that reason, we call them dural sinuses. So the superior sagittal sinus is a dural sinus. So is the transverse sinus. It too is a dural sinus. So these large veins pick up this fluid Of course, it's maybe full of waste products now. The nutrients have been depleted, and we're going to take that back toward the heart. So CSF is reabsorbed here in these little arachnoid villi that you can see sticking up and back into those dural sinuses. In your course pack on page 112, you can find the detailed flow of cerebrospinal fluid. I highly recommend that you sit with a picture like the one we just went through and work your way through this flow pattern. This flow pattern will be on both lecture and lab exams. So if a picture doesn't help, then flashcards may help you to get this committed to memory. You're looking here at a picture of hydrocephalus. Hydrocephalus can result when there's excess cerebrospinal fluid in circulation. Perhaps it's not draining like it should. Now, in babies, because their skulls are more flexible, the skull will actually expand to accommodate this extra fluid. So you can see in the picture here on both left and right, there's a pocket of fluid in the baby's head on the left, and the head is swollen to take on the extra fluid in the right. Babies have fontanelles. You may recall that those are skull parts that have not yet fused together. After about age 1 or 2, those skull sutures have formed, and the head is no longer flexible. That means that after age 1 or 2, if there's excess cerebrospinal fluid, that extra CSF can cause brain damage. Here, this baby may be fortunate enough to escape brain damage because of the flexibility of those skull parts. Here you can see what hydrocephalus does to the brain. On the left, let's go ahead and look at the lateral ventricles. I'll circle those for you. These are lateral ventricles here and here. You might recognize that diamond-shaped fourth ventricle. And on the right-hand side with hydrocephalus, you can see there's so much fluid that we've flooded the inside of the brain surface. In fact, there is very little space outside the brain between the brain and the skull, all along here as well. So this can cause brain damage. It can also shove the brain down into the foramen magnum and shut off functional brain control. Most of our conscious thought and processing occurs in the outer 1/8 inch of your cerebral cortex. So that means if we have extra fluid out there, it can actually crush that brain functioning. Let's now turn our attention to the blood-brain barrier. It is still found on page 112. Now, there are capillaries that serve your brain. We know that those capillaries can help create cerebrospinal fluid, but we may not want everything in those capillaries to be able to infiltrate your brain tissue. So we have this set up in the brain that's special to the brain, and it's called the blood-brain barrier. This blood-brain barrier is a set of capillaries that prevent most items from traveling into the nervous tissue. How do we do that? Well, there's two main things that are at work here. We have tight junctions between those cells, and we also have astrocytes. These tight junctions located between the cells prevent fluids from traveling between the cells and into the surrounding nervous tissue. Astrocytes perform a lot of that same function. They perform an impenetrable barrier between the cells so that fluids and other items do not get into the brain tissue. Here you can see those astrocytes. They are somewhat star-shaped cells, so they are part of the formation of the blood-brain barrier in the capillaries of the brain. Now, the benefit of the blood-brain barrier is that not everything can reach the brain. Bad things like viruses, for instance, may have a more difficult time reaching the brain or spinal cord tissue. But there's also a disadvantage here. Certain things are able to reach the brain, like alcohol, which may be a bad or a good thing. But of course, certain medications that we would like to provide for the brain, perhaps for cancers, for instance, we cannot get to cross that blood-brain barrier. So in order for items to pass across this blood-brain barrier, they must be lipid-soluble. Now, there's only two areas in the brain that you'll want to make note that are missing the blood-brain barrier. That's the medulla oblongata, because it houses a vomiting center, and the hypothalamus, which monitors the chemical composition of our blood. Everywhere else in the brain has a blood-brain barrier. Let's now turn over to page 113 and talk about the regions of the brain. There are four primary regions. We have the cerebrum, which is this mushroom-shaped cap that forms the outer surface of your brain. We have this cerebellum at the back part. We also have the diencephalon, The diencephalon includes the thalamus, which is shown right here, the hypothalamus, which is sitting right nearby, and the pituitary gland. So all of that is part of the diencephalon. Here they call it the forebrain. Okay, so diencephalon is technically thalamus, hypothalamus, and pituitary gland. The brainstem includes 3 components. The midbrain, which is here. There's also the pons, which is this piece that kind of sticks out here. And also the medulla oblongata, which sits a little further down. So these are the main parts of the brain. We're going to talk through these not only on this PowerPoint narration, but on the next one as well. So let's talk about the lobes of the cerebrum. We know the cerebrum is the outermost wrinkly portion of your brain. The cerebrum has lobes that correspond to the names of the skull bones. So if you remember your skull bones, then you're good to go here. First of all, let's find the frontal lobe here. We also have the parietal lobe, corresponds to the parietal bone. We at the back of the brain have an occipital lobe and a temporal lobe over by where the ear is located. There's also an insular lobe, which we can't see from this view, which includes deeper tissues to the cerebrum. So the cerebrum functions in consciousness, perception, communication. This is where we perform our speech. Our understanding and logic occurs here, and our voluntary movement occurs here as well. There are interneurons in the cerebrum that also integrate or associate actions between sensory and motor neurons. This is where our executive decision-making functions occur, is in the cerebrum as well. So let's talk about the white matter and the gray matter in the brain. In the picture down on the bottom left, you'll notice the gray matter actually forms the superficial portion of the cerebrum. The white matter forms the deeper portion of the cerebrum. You'll notice in this picture that we have little shallow grooves here called sulcus, and we have raised hills called gyrus. Okay, so religious or not, I had a student years ago come up with a great way to remember gyrus are hills and sulcus are grooves, and I'd like to share that with you. She said that God was up and Satan was down, so G for gyrus is the hill that points up, and sulcus is the groove that points down. If you've got a really deep groove, you'll notice in this picture it's called a fissure, but keeping those straight here will help you understand what we're going to talk about a little later, which are precentral and postcentral gyrus and things of that sort. Here's another view of those 4 cerebral lobes: frontal, parietal, temporal, and occipital. They are named and associated with those skull bones. Like I mentioned earlier, there is an insular lobe, which is deep to the brain. This insular lobe is pictured in here, and we've talked about some of those components already, and we'll do so later on as well. Note again, the gray matter is the outermost portion of the cerebral cortex. It makes up the outer 1/8 inch of our brains. There's also white matter deep to that gray matter. If you look further into the brain, though, you'll notice that there are deeper pockets of gray matter down here. Some of these nuclei deeper inside the brain play an important role. We'll talk about some of those, but those deeper pockets of gray matter are also found here in addition to the white matter. Now, thinking about the spinal cord setup, this is actually reverse of how the spinal cord's gray matter and white matter is established. Another closer look of the gyrus and the sulcus. Keep in mind that gyrus is the heel of tissue and the sulcus is the groove. Another way to keep this straight might think of a sulk. You know, if you're sulking, you're sad, you're down in the dumps. So a sulcus is a grooved portion of the brain. And like we talked earlier, a fissure is a deeper sulcus. So let's talk about some of the functional areas of the brain. You can see here that we're going to talk about speech, taste, smell, hearing. We're going to talk about where vision is housed, where hearing is housed, motor control, things of that sort. So I did want to go ahead and point out to you a couple of important parts. You can see the precentral gyrus is marked here as this hill of tissue right here. There's actually a postcentral gyrus here. I'll go ahead and label that one for you. And it's found back here. There's a groove that separates the two. It's not a deep groove. It is simply called a sulcus. And so that central sulcus separates these two gyruses from one another. A lot of these areas of the brain that we're going to discuss are called Brodmann's areas. These are functional regions of the brain. All right, and to finish this up, let's talk about fiber tracts within the brain. This is an area of white matter in the brain. This is specifically in the cerebrum, and these fiber tracts provide connections. Now, before we go any further, you may have heard the word tract before, and that's because it's a collection of neurons in the central nervous system. The word tract tells me I'm in the CNS, not in the PNS. We're going to talk about association fibers, commissural fibers, and projection fibers. So let's first of all tackle those association fibers. They are found in one hemisphere of the brain. They travel the length of the hemisphere from front to back or back to front. So they run the length of the brain's hemisphere. So association fibers connect point A to point B inside of one hemisphere. Commissural fibers are things that run across. So this corpus callosum that you see here is actually a commissural fiber. It connects left and right hemispheres together. Here's another one, anterior commissure. So fibers that run from left hemisphere to right hemisphere and vice versa are called commissural fibers. Finally, projection fibers travel from the brainstem up and out. So you can see here projection fibers come from the thalamus and they are projecting literally information up here to the cerebral cortex. So let's say that you put your hand on a hot stove. The reflex jerks your hand away, but ascending tracts called spinothalamic tracts take information to your brain. That information arrives here at this thalamus, and the thalamus relays that information onto the cerebral cortex using projection fibers. So here you can see the corpus callosum a little more up close. This piece right here is a white fiber tract. It connects left and right hemispheres together. This is critical, especially if somebody doesn't have two functional eyes or two functional ears. It allows for things like vision and hearing to travel across this corpus callosum and into the opposite hemisphere. That's necessary because that may in fact be where processing of that information occurs. You can see again here the corpus callosum connects left and right hemispheres. And another nice view of how these fibers work. You can't see in this picture any association fibers since they run the length of the hemisphere, but we do see commissural fibers that connect left and right hemispheres, and we see projection fibers that travel up from the spinal cord. Here they come, they travel through the thalamus region, and then they project information onto the cerebral cortex.